All right, well, welcome everyone. This is our second webinar in a series for the California Psychedelic Bar Association. I should note that it's being recorded and will be posted on YouTube, and if we can figure it out at some point, some podcast apps. I am Paul Slattery, one of the two co-founders of the California Psychedelic Bar Association. When I'm not doing this, I am the general counsel of Eleusis, which is a psychedelic pharmaceutical company. My co-founder, Rebecca Lee, can't make it today. She is the general counsel of Journey CoLab and will be doing some of the future interviews. Our previous webinar was with Professor David Nichols, which you can find on our YouTube channel. This webinar will continue to lay the foundation of understanding the science and the research efforts in psychedelics before we turn to more strictly law and regulation. We're very lucky today to have Professor Charles Nichols. I should note that he and I work together professionally, so we know each other pretty well. In addition, Professor David Nichols from the prior webinar is Charles' father and gave me express permission to call Charles Chuck. But I'm going to try to avoid doing that. So hello, Professor Nichols. How are you today?
Oh, okay. Good. Yeah, you could either Charles or Chuck. There's an interesting story behind that.
I think I'll stick to Charles. I don't want to please your dad too much here. So you have a very long resume, and I'll pull out a few parts of it, but it'd be sort of boring for me to just rattle off a summary. And I thought it'd be more interesting for you to tell sort of your story and how you see it. So can you take a few minutes and just sort of walk us through your career path from grad school to now doing psychedelic research?
Sure. First, apologize in advance. I've had a cold recently, so I sound really funny. And but yeah, I've a lot of people assume I got into psychedelics because of my father, and that's like not the case. I wanted to do something completely different. From my parents, both my parents are pharmacologists. I started off at Purdue University in chemistry. I found chemistry pretty boring, so I switched to biology. And this was in the mid to late 1980s. And so the first PCR machine had just come out, and people were able to do molecular biology. And I started doing undergraduate research in the bacteria genetics lab, and I found that really fascinating. Doing that. So when I got my undergraduate degree, going off to graduate school, I didn't really know what I wanted to do, but I know I wanted something to do with genetics. So I looked at a few different programs, got accepted to a lot of different places, and ended up going to Carnegie Mellon because they had a developmental biology. Well, their biology program there was pretty heavily focused on developmental biology, but incorporated a lot of genetics. And they were using a lot of diverse organisms, from mice, flies, yeast, sea urchins. So I thought, oh, that would be a really great place to go. Still didn't know what I wanted to do. And it was, I had to pick labs to do rotations. And it was in the first few weeks, and I would see all the professors come out and give you the dog and pony show. This is what we do in our lab. And this one young investigator, John Pollock, showed these beautiful pictures of neural networks in the eye and brain of the fruit fly that he had taken using fluorescence microscopy. And he was a physicist by training who had done his postdoc in Seymour Benzer's lab at Caltech in fruit fly eye vision and retina development. I thought, oh, that's really cool. So I got my PhD studying the developmental genetics of the retina of fruit flies and really learning hardcore genetics, molecular biology, developmental biology, neuroscience. And after several years of working with fruit flies, I thought, no, this isn't where I want to be the rest of my life, because even though the conceptually was very interesting, but the practical day-to-day experiments were grinding flies up, extracting their nucleotides and running them on a gel, and looking at black and white spots on a filter paper essentially all day and trying to interpret that, which conceptually was interesting. Just really, I wanted to do something more behavioral related. So really no idea yet where to go. And I was coming close to graduation and looking at postdocs. And I had accepted a postdoc in a laboratory doing Parkinson's disease. And I saw, was waiting for my ride that one day to come pick me up, and they were late, and a little embarrassing to tell this. I had a desk chair that spun really well. And so what helped me think a lot of times was I would sit at my desk chair and sort of spin around. And so I was waiting and I was thinking about things, and then it stopped. And when it stopped, my eyes landed literally on the desk behind me, the other graduate student in the lab, on a big cluster hire advertisement for Vanderbilt Neuroscience. And like, oh, okay. Still thinking about postdocs, looked it up, and a lot of really interesting stuff there. And I noticed the name Elaine Sanders Bush was listed. And I had recalled my father talking about Elaine Sanders Bush, saying that she was a really good scientist. He had collaborated with her on a couple of projects with serotonin and pharmacology. I sent her, oh, cool. So I sent her an email, and like very promptly got this very short reply. No, I don't have any funding. I'm not sure why they had me on that list. I'm not looking for a postdoc. It's just like, okay. I was all set to go to this other postdoc, and it was a month or two later, I think, several weeks at least. She sent me a follow-up email that, oh, I've got money now, and I've got a project in mind. If you're still interested, can you come out and interview next week? Like, oh, okay. So went out to Vanderbilt, saw the lab, met her. She was great, really fell in love with the environment, the people there. And the project was looking at effects of gene expression, of LSD on gene expression in the brain in rats. And I thought, oh, that's pretty cool. So she was looking for somebody who knew molecular biology and genetics to kind of bring her lab into that era, from sort of a classic receptor pharmacology, membrane binding signaling, and be able to help train other people to do molecular biology. And I remember when I gave my interview, like half the people in the lab fell asleep during my seminar. I'm like, oh my God, I'm blowing this. But she offered me the position, and turned down the other offer, went to Vanderbilt, and it was about a month or so later, she came out of her office. She was holding my CV, and she says, I see, I just noticed now that you went to Purdue, and I know somebody at Purdue. Do you know Dave Nichols? And I said, well, yeah, actually, he's my father. And she just was very excited at that. Like, oh, wow. So from then on, half the time she called me Dave around the lab. But that was kind of my start in the world of psychedelics back in 1997, was in Elaine's lab looking at gene expression from psychedelics in the rat brain.
Very cool. How did you then wind up at LSU doing research you do now?
So I ended up at LSU, again, another twist of fate. I was on the faculty job market. My father had counseled me that trying to find a job in psychedelics was the career killer. It was like the kiss of death. Said, don't do it. Nobody gives a darn about psychedelics. And I was stubborn. I persisted. All my CVs, I'm studying 5-HT2A agonists. I was using fruit flies again at this point, because a couple years into my postdoc, a paper came out where they were giving fruit flies methamphetamine and cocaine, and then looking at gene expression changes. And the little light went off. Oh, you can do behavioral pharmacology in fruit flies. So I got some fruit flies from down the hall. I gave them some LSD, did a little project looking at how LSD changed visual perception in the fruit flies through 5-HT2 receptors, and started to look into serotonin receptors in the fruit fly, and found that they weren't really characterized. Expression wasn't known, function. So started to develop a fruit fly as a model to study serotonin neuropharmacology. So when I went on the faculty market, it was studying 5-HT2 receptor pharmacology in mammals and in fruit flies to try to understand how serotonin 2A receptor activation influences behaviors. but it was still, trying to find the mechanism of action of psychedelics. And I was applying to mostly neuroscience departments, pharmacology departments, but nobody wanted people working on psychedelics. And then I saw one ad for LSU, the Health Sciences Center. And I looked at the department. It was full of a bunch of cardiovascular and kidney researchers. I was like, oh, it's not a fit at all. And I printed it off and I threw it in the garbage. It's like an hour later, I remember I picked the application out of the garbage can. I said, you know what? It's New Orleans. I love going to New Orleans. The music, the food, I don't care. I'm going to apply. So I applied, sent it off back in the snail mail days and my application. And it was about 2 weeks later, Elaine came and said, oh, I was just talking to the chair of the department at LSU, Steve Lanier, and they're really interested in your application. And it just turns out that Steve Lanier was going to be at LSU the following week for a symposium for Lee Limbert, who was the former chair at Vanderbilt. And I talked to him there for a little bit. And the following week, I was interviewing down at LSU and given an offer. And as it turned out, LSU was at the time a young, vibrant department, lots of funding. The chair saw me sort of positioning between the molecular group and the behavioral group, because there was a lot of drug abuse research that was going on there, looking at different models and a lot of cell biology and genetics, and really liked the environment, the people down there. So I took the job and I've been there for 18 years. Yeah.
Great. Okay, yeah, let's move a little bit into psychedelics. The first question, so I've actually noticed more variability among researchers in how they define psychedelics than I so what's your definition? How do you sort of capture the class? And just because folks have likely heard of it in the space, given it's available for prescription now,
right.
Can you distinguish ketamine, or if your definition distinguishes it?
Right. I think for many years, any kind of drug that changed your perception of reality, distorted sensory perception, was called a hallucinogen. And so for several years, if not decades, drugs like LSD, marijuana, PCP, basically, even any drug that could change your perception was lumped under the category of hallucinogen. So that kind of continued through the 80s and the 90s. And then when there was Humphrey Osmond coined the term psychedelic in several decades ago to really kind of distinguish the effects of LSD and psilocybin and mescaline from the other hallucinogens, because they had this subjective difference. And so they had psychedelics kind of loosely then associated with hallucinogens, and some things that were hallucinogens were called psychedelics, but they weren't, something like PCP. So there was a need that was recognized to kind of pharmacologically distinguish all of these different types of hallucinogens. So the modern pharmacology definition of what a psychedelic is a drug that produces its behavioral effects, its changes in perception and reality through activation of a protein called the serotonin 2A receptor. So the other types of hallucinogens like ketamine and PCP, those are primarily acting at blocking a protein called an NMDA receptor. And those are in a category of dissociative anesthetic. And then cannabinoids activate the CB1 and 2 receptor proteins to produce their effects. So that's a different mechanism. So a drug like MDMA, there's been a lot of back and forth on, is it a psychedelic or not? It doesn't have any affinity or effects at the 5-HT2A specifically, It has indirect effects through causing a release of serotonin that activates all the serotonin receptors. But its primary mechanism of action is not 5-HT2A receptor activation. So from a pharmacological standpoint, MDMA is not a psychedelic, and it's not really a hallucinogen because it doesn't produce hallucinations. So my father coined a term called entactogens that really defines that class of drug. And I think a lot of people, most people in the field refer to MDMA as entactogens. And some people refer to them as entheogens, but that doesn't quite really cover the same meaning as entactogen does. So from a pharmacological standpoint, there's fairly clearly well-defined barriers of what these drugs are, but I think it's in the lay person's perception because there's a lot of overlap in the effects of what they do at the sensory level. But from a pharmacological standpoint, they're very distinct.
Perfect. So our previous webinar covered this a bit, but I'll ask you too. What are the most promising combinations of psychedelic compounds and therapeutic indications you see being pursued? Can you give us sort of an overview of the therapeutic research?
Yeah, well, you know, the lion's share of it's been focused on psychiatric disorders, initially with depression and anxiety associated with cancer or other life-ending events. And now that's been extended to depression, major depression, treatment-resistant depression, anxiety. So that's the lion's share of clinical studies now, but there are some interesting studies now, I think, looking at, for example, migraine headache, some of the studies that are going on at Yale looking at the potential use of psychedelics to treat migraines, which may invoke some of the anti-inflammatory effects of psychedelics. There's also some OCD was an older indication, actually one of the first that was tested in the clinic, but I think that's one that I think deserves some more clinical attention. There's so many trials now going on. There's some going, you know, for Alzheimer's, for Parkinson's, neurodegenerative disease. It seems like, you know, there's lots of feelers going out in different directions. And I think some of those will get traction and some of them won't. It just depends on really what the science informs us on some of these pilot clinical trials that are going out for the different disorders.
So we have a pretty broad base of folks who may be watching. So there's some like me, who are general counsel of the psychedelic pharmaceutical company, or patent lawyers who are pretty sophisticated about pharma research, but also probably folks, you know, in the decriminalized psilocybin market or criminal law or whatever else. So I suspect not everyone has a sense of the basic process of pharmaceutical development. In other words, of what you do all day. So could you sort of walk us through how that's conducted, you know, discovery, screening compounds, election of a lead, rat models, that kind of thing?
Right. Yeah, there's two main ways. There's like big pharma drug discovery, which is kind of brute force. And then there's smaller biotech pharma and academic drug discovery, which is more intellectually and rationally driven. So what most of the psychedelic firms now are sort of focused on, they're the smaller biotech. None of the larger pharma companies have really so far put their feet into the psychedelic space. But in general, the process is the same. You identify the relevant disease or treatment modality, in this case, activation of 5-HT2A receptors to produce a therapeutic effect. The traditional way would be to identify what the primary receptor target is. For high blood pressure, for example, that could be the beta adrenergic receptor. For psychedelic drugs, we know it's the serotonin 2A receptor. So you can, in one approach, screen physically hundreds of thousands, if not millions of chemicals just to see if they activate the serotonin receptor through these big robotic screening devices. That's what Big Pharma and DARPA has actually funded Brian Roth's lab several tens of millions of dollars to do that kind of screening with the 2A receptor. But his screening approach that they've taken is this in silico virtual screening, and they've screened several billion molecules virtually in a computer program, model of the 5-HT2A receptor to predict which ones will activate it. And then they've made certain ones of those and look for activity. And what I do in my laboratory is I take sort of existing molecules, looking at their pharmacological properties, what they do in cells, what they do in animals in a particular disease model, or looking for a particular signaling molecule pathway. And I look to see how I can potentially modify that molecule to change an aspect of that molecule to enhance its activity in the direction we want to go. Like if I may use the example of the drug that Elucis is developing at an overall level. For that drug, ELE-102, what we did was we identified a particular drug that had an unexpected activity in one of our assays that gave us a clue that it might be a potent activator of the 5-HT2A receptor, when based on the literature, you would think that maybe it wasn't. So then looking at that molecule, we looked at how could we modify that to increase, potentially maintain and increase activity of that molecule, but decrease its behavioral effects, because we know certain ways that we can modify the parent molecule that would result in a decrease in behavior based upon the work of people like my father. So we made 2 changes to this molecule that fortunately bared fruit, that it stayed anti-inflammatory and it was induced less behaviors. So that's what you call rational drug design. When you pick something, you figure out how you can try to modify it and make those modifications to see if you can validate your hypothesis of those modifications rather than the brute force approach. So once you go through either one of those brute force or rational approaches, you have candidates. So then you have candidates that are usually identified through cell-based screening. And that's how we identified ours with the rational approach, and how Big Pharma and how other labs in silico, you end up with candidates. The problem then is, how do you know if it's even effective in a disease model? So then you have to move to an animal model of a disease and check it in an animal model. And that's where most drugs fail. That they work great at activating the receptor in a computer or in a cell to turn on a particular protein, which will make light that you can detect as a reporter. But then when you give that drug to an animal that has a disease that you're predicting changing the activity of that receptor will modify that disease, that's where probably 3/4 of drugs fail at that level. So the big pharmaceutical approach is to throw, 20 different drugs into an animal and look for one that kind of works, and then take that molecule and tweak it with medicinal chemistry, throw it back in the model, and see if it has performed better. The other way that most drugs get killed are that they're toxic to the animal. They look great in cells. You put it in the animal and it treats whatever the disease is, but it has this off-target effect that maybe is cardiotoxic, is toxic to the liver, toxic to the kidneys. The most common one of these is a protein called the hERG protein that causes fatal arrhythmias. So a lot of drugs now can be screened out now before you go into an animal, but toxicity off target is what kills the rest of the drugs. So like 90% plus or more of drugs that make it to an animal model fail due to one reason or another, the toxicity, or they're just not efficacious. So once you get to that level and you show it's safe, then you go to the FDA, say, we have a safe drug. It's not toxic. You have to spend millions, tens of millions, usually, on a drug to show that it's not safe. And so you'll have one or two that will have survived a 500,000 drug initial screen, will be pared down to one or two drugs that you show are safe and are efficacious in more than one animal model. You present that data to the FDA, requesting approval to do a phase 1 safety trial in humans. And then that's where the majority of the rest of the drugs fail. They give it to a human, and either in the phase 1 trial in the human, it produces a toxic effect of some sort, or in an early efficacy trial, it just doesn't work. So the number of drugs that are actually small molecule drugs that are making it to market every year, the number is about 20 to 30 out of the billions that start off every year to go through that process. So most major pharmaceutical companies, at least in the terms of the neuroscience space, have gotten out of that space. There have been no new major discoveries in the field of neuroscience, depression, things like that, since essentially SSRIs were discovered back in the 1980s. So they figured it's just a losing proposition. So they've now the model is let a biotech company, a smaller, more nimble company, come up and do the discovery. And then when you're at a certain phase, you can partner with one of these larger companies that can then put in the hundreds of millions to do the larger scale trials. The first phase 1 trials or phase 2 trials will use like 20 to 100 individuals, and you might see efficacy there. And that may be promising, but you may have some selection bias of who you've got in those trials. So you need to go to a phase 3 trial, which is several hundred individuals at several different sites, sometimes around the world, to show that it works in everybody. Those trials cost hundreds of millions of dollars that take several years. And if you make it through that whole process, then you've got probably a blockbuster drug, which gonna sell billions. It's just a really long and arduous path to get from concept to market. And the road to that is just littered with failed drugs and companies. And so it's really difficult. It's really difficult.
It's really helpful. So focusing on psychiatric indications first, and then we'll get into anti-inflammatory stuff in a minute. What do we know about the therapeutic mechanism of action for these compounds? Do we sort of understand how they work, if it's peak plasma concentration or area under the curve, or do we know?
No. Nobody knows how they work. There are some interesting clues at this point. We know that it requires activation of the 5-HT2A receptors. Pretty much, if you block 5-HT2A receptors in humans, you block the behavioral effects. In animals, it blocks the behavioral effects. So at the fundamental level, we know the mechanism involves activation of serotonin 2 receptors. But what downstream beyond that is still an open question. There is some evidence now from the imaging studies on how brain network connectivity is being changed. And that's kind of like looking at roadmaps. Looking at a roadmap doesn't tell you how a car really runs, but it kind of tells you important areas that may be involved in the therapeutic effect within the brain, and how those connections might be important. There's some evidence that increased synaptic plasticity may be important, but that still hasn't been really reliably shown yet. There's some very intriguing preclinical rodent studies suggesting that, but still that's not known. If it's exposure, if it's area under the curve or peak plasma, it appears for therapeutic effect, I would say it's more likely, in my opinion, peak plasma levels rather than area under the curve. We've got some animal data that suggests that, and there's some human imaging data from Gita Knudsen's laboratory looking at receptor occupancy, showing that when you hit that receptor occupancy of 60% of the receptors, then that triggers sort of the peak experience. And the peak experience is what's the most highly correlated with therapeutic effect. Although in humans, the time that they have to undergo, they have the peak experience, but they're still hallucinating for several hours after that with psilocybin. So the big open question now is, can you shorten that? And based on some of my work that we've been doing with psilocybin and different routes of administration in rodents, it appears that it's more shifted towards peak plasma rather than area under the curve.
Sort of a related question. I'll ask about microdosing, just because if you look at the popular press or wellness trends, or if you're a young professional wandering around Venice, you'll find plenty of discussion of microdosing psychedelics. There's a little bit of pharmaceutical development there, but not much. And there are probably regulatory and practical reasons for that, with folks taking things home and things like that. But can you give us a sense of what we know about the effects of microdosing? What's the science there, if there is much?
There's very little. There's a handful at best of studies that have used, rigorous blinding controls, known substances for microdosing studies. I think there's really only two groups that have looked at it empirically. One of them was the ELUSA study, and the other one was a couple publications out of Harriet DeWitt's laboratory in collaboration with Katrin Preller. And the other studies have all been really kind of survey studies, naturalistic studies, where they're asking people essentially to microdose and tell us how you feel. And the problem with those is there's no, you don't know what's in the drug that they're taking. They don't know what that drug is, how much they got. They just know that maybe they didn't feel anything. But what some recent publications have shown and suggested is that the perceived positive mood and cognitive enhancing effects of microdosing psychedelics is due to expectational bias. Put simply, a placebo effect. They're expecting that the microdosing is going to enhance their creativity or their feeling of well-being, and so it does. The studies that have looked at objective laboratory measurements of people who have taken low-dose subperceptual psychedelics in these two different organizations that have published on it have shown that there's really not really a measurable effect in any of those areas. But what Harriet DeWitt's group did show was that at this lower level, it was changing some aspect of network connectivity. So the low levels of LSD, and that's what these two studies or two groups were using, it appears that the low levels of LSD are doing something to the network connectivity and function. But at least in the measures that people are looking at, like cognitive enhancement or changes in perception, things like that, feelings of well-being, that those aren't being picked up. The sensitivity is not there. So maybe other outcome measures could be looked at, but the evidence is there that I think there's something that's going on, but it might not be enough to affect any changes that are really measurable. And that's with the psychological side of things. With the anti-inflammatory side of things, microdosing might give enough of certain psychedelics that would have an anti-inflammatory effect that would be therapeutic, because the effect of that is really super potent.
Why don't we just jump to it? So this is really a signature question for you. I suspect most folks watching are aware of the psychiatric research, but they may not be familiar with therapeutic applications outside of sort of psychiatric indications. And you are a pioneer of potential use of psychedelics for anti-inflammatory therapeutic indications. Could you speak about sort of the applications of psychedelics outside of psychiatry? And folks may need the background of the 5-HT2A receptor being expressed throughout the body and things like that.
Oh yeah. So, when you think of psychedelics, you think of course, visual hallucinations and the trip and things like that. They're psychedelics. So we, I won't say accidentally, but serendipitously discovered the anti-inflammatory effects of psychedelics when we were looking for some cell-based models to study the effects on gene expression. I didn't want to have to keep taking the brains out of animals to do the analysis. And if we could have some meaningful cell-based systems to look at some of these gene expression changes, that would have been advantageous. So we did a whole panel of different types of cell-based assays. And through a random circumstance of events, again, that depended upon one of the events necessary for the discovery was Hurricane Katrina. So without that, I don't think we would have discovered it, because that allowed a postdoc who was in pathology, who had a specialization in 5-HT2A receptors and hypertension from a study, his PhD doctorate, to not want to move with his mentor to another university. He wanted to stay. So he moved over to my lab and brought his skill set of looking at vascular inflammation to my lab. And so we put together an assay where we looked at vascular cells in a cell-based model of atherosclerosis, and found that psychedelics were super, super, super potent. They are about 500 to 1,000 times more potent as an anti-inflammatory in these cell-based models than they are at inducing the effector pathways to produce behaviors. And we have looked at several disease models now. We've looked at disease models of cardiovascular disease and atherosclerosis in mice. We've looked at asthma in both mice and in rats. We've looked at models of irritable inflammatory bowel disease in mice. And the one we've used the most is the asthma model. And in that model, it's allergic asthma, seasonal asthma. You're triggered with pollen or an allergy, and you've got inflammation, your bronchioles close up, you've got mucus, you can't breathe. You take an inhaler, and it directly causes the smooth muscle cells to relax. It's not disease modifying. Your inhaler for asthma just causes your smooth muscle cells to not be constricted and relax them so you can breathe better. But what we found, what psychedelics do, is it's disease modifying. It reduces the pulmonary inflammation. It prevents and reverses the thickening and the hardening of the bronchioles, enables easier breathing in our animal models, about 30 to 50 times lower levels than necessary to be the threshold behavioral level. Like for our prototypical drug, which is called DOI, for behaviors, our DOI specifically is what we use. For a behavior in a mouse, it's around 0.1 to 0.3 milligrams per kilogram is the minimal necessary to detect a significant change in in behaviors. And in our asthma model, it's not 0.1 to 0.3, it's 0.005. So that much more effective. So we've seen similar potencies in the other animal models. So if translated to human biology, that would suggest that DOI, which have to take around similar potencies, you would have to take microgram amounts of something like DOI in humans as an anti-inflammatory. And so the drug development that we've been doing in my lab has been to kind of engineer out that behavioral effect. And so our lead that's come out of my lab for that hopefully will even further separate that. But several different models. My collaborator, Dr. Tim Foster, has been looking at models of ocular inflammation, retinal inflammation and disease, and in collaboration with Elucis, been also exploring, the ocular and retinal inflammation, and there's a very potent effects there as well. So it's not just one organ, one system, and we don't know what other models would show us a hit, because the target, the serotonin 2A receptor, is in every tissue, nearly every tissue in the body. It's the most widely expressed serotonin receptor. What it's doing so widely expressed, you'd think it would be absolutely critical for survival. But if you make a knockout of that receptor, it's actually amazingly normal. So it's more of a modulator that, when the system is perturbed, doesn't allow the system to respond properly. And I think with one of the things might be with the inflammation. So we're activating the receptor differently than serotonin. And so where serotonin is activating the receptor to produce inflammation, like if you give serotonin in these models, it'll actually cause asthma. Our drugs and psychedelics, but not all of them, LSD is really bad at this, at activating the receptor. So it engages different amino acids in the binding pocket that produces a slightly different conformation that inside the cell, instead of making an inflammatory response, it makes an anti-inflammatory response. And it's really sensitive to this anti-inflammatory effect.
So this is a broad question, but it's a sort of choose your own adventure, however you like. How has performing research in psychedelics changed since you started? What's different about doing it today than sort of your earliest experiments at Vanderbilt?
When I started back in '97, which was 25 years ago, there were probably about 6 people in the world who were actively studying psychedelics or 5-HT2A receptors. And it was all in terms of potentially understanding schizophrenia or psychosis mechanisms better. There was the scheduling, the barriers to doing research over the last 40 years, since 1970, when all the human research studies stopped. It was very, very difficult for anybody to get the necessary permission to do these kinds of experiments and studies. So for somebody who wanted to break into the field, it was next to impossible to do that. My postdoctoral mentor, Elaine Sanders Bush, had been studying psychedelics since the 1960s, when she actually volunteered for an LSD study and had a horrible trip. She said it was the worst experience of her life, but it was the one time that she took psychedelics, and she talks about it in the class that she gives. But how such a small amount could such profoundly change somebody's perception, just so much fascinated her, that she started doing basic science research in psychedelics back around the late 60s, early 1970s, and had a series of discoveries. So coming out of her laboratory, I was able to continue that on and use my provenance of research in the field to facilitate getting my DEA license. And still, that took almost a year to get everything in place to be able to use Schedule I drugs. And I think it was just too onerous for most people to want to try and to do that. So when I got into the field, maybe 6 people, and as my father told me, it was a kiss of death for my career if I tried to do it, because nobody cared. And then I got traction with it. I was able to get grants. I was studying serotonin and psychedelics in flies to study serotonin systems. And then when we hit the anti-inflammatory effects of psychedelics, was able to secure funding and grants on studying anti-inflammatory aspects of 5-HT2A, but it was all really 5-HT2A receptor function. It wasn't psychedelics. I don't think you could be taken seriously until 3 or 4 years ago by saying, I'm a psychedelic researcher. I study psychedelics. Because it would be when I was a postdoc and even a junior faculty, yeah, I study the effects of psychedelics. They're like, oh, okay. How's that working out? You're giving LSD to fruit flies. And now I think there's really been this paradigm shift in society that, you tell people now you work on psychedelics, and like, oh, that's really cool. So it's no longer viewed as this brain melting class of drugs that's going to destroy your life and set you on the path of abuse and destruction. That people really understand now that there's legitimate medicinal value to the study of these. So over the last 3 years, just the amount of publications on serotonin 2A receptors has mushroomed. And the DEA is trying to streamline the process to enable new researchers in the field. So if they have, they presented some data at the Psychedelic ISRP Society that we formed a couple of years ago, showing the number of Schedule I licenses that they've issued over the last couple of years. So they're slowly coming around to sort of facilitate that. So there's a lot more investigators in the field. When we had our ISRP meeting here in New Orleans, my father said he doubted initially if more than 50 people would register to show up when we were in the initial planning stages in like 2017, 2018. And so we booked the room in the conference to accept 125 people as a maximum. And when it came time for the conference, we probably could have had double that, as in, real scientists, MDs, PhDs, graduate students studying psychedelics and 2A receptors. And now it's even so much more. So it's mind-boggling how far just the public perception has shifted and how quickly it's shifted. And with that perception, research into psychedelics.
Yeah, so I'll get to sort of what's challenging, what could be better in the regulation of psychedelic research in a second. But I think folks may not know, could you walk us through first just what it takes to do research on these compounds from a regulatory perspective, Schedule I license, what it's like to have one, procedures?
It's different depending upon who or where you are. I can give you the example of my university, for example. Some universities will have a campus-wide licensing, but that's pretty restricted to some of the few in the major universities. Most universities, each individual investigator has to get their own license from the DEA. So I, as an investigator, have to put together a package to send to them that contains essentially my CV, my proposals, what I want to work on, the drugs I want to work on, why I want to work on them, how much I need. I have to get a state license. I have to get a federal license. Each is dependent upon the other. So I contact the state. You have to contact the state, tell them that you're seeking the federal, and then they get everything ready and tell you, okay, let me know when you have the federal number, and then we'll give you the state. You apply to the federal, and that often involves sending in approved animal protocols. Like, these are the experiments we want to do. These experiments have been approved by our local animal use and care committee. They've been approved by, if you're doing human studies, the local IRB, biosafety committees. They then send those off to the FDA for evaluation to inform the DEA that these experiments seem reasonable. They evaluate the amount of drug that you're requesting. And then if all looks good there, this process can take weeks to months. Then they have to send local field agents from both the federal DEA and the state DEA to come inspect your facility. They look at your safe, the security of where your drug safe is kept. And when you have 2 DEA agents with sidearms show up at your lab, and badges. It's okay. So if they look, everything looks good, then they report back, and then depending, weeks or months or days, depending on, they then will give you your Schedule I license. So I've heard examples of it taking for over a year, to the fastest I've heard it has been 2 to 3 months. And so the DEA is trying to streamline it from on average maybe 9 months to something a little bit less onerous. But it's still, there's a lot of paperwork, there's a lot of agencies involved. And then once you get your license, then there's the annual renewal. So if you want to add a drug or add even an experiment, a different experiment that you want to do, you can't just go into the lab one day and say, oh, I think I want to do this experiment with LSD, which is different than what I have been doing. You have to write up a new protocol, get it approved, and then submit that for the DEA, get DEA's approval to use the drug in that experiment. So the license to use a Schedule I drug isn't a blanket license that covers everything in your laboratory. It's a number that allows you to apply for permission to use it in specific experiments in your laboratory. And they will do random inspections of the laboratory where 2 agents will just show up at your door and say, let us see your logbooks, where they can then proceed to look at your inventory, weigh your inventory, check your running logs, check your security, make sure everything's okay. So these random inspections will also happen from both the state and federal. So it's not for the faint-hearted to do research with Schedule I. And Schedule I includes opiates, psychedelics, cannabinoids. I think most of the Schedule I research is now, it's cannabinoids and opiates, but psychedelics are increasing.
So recently the DEA has been engaged in some rulemaking proposals to schedule additional non-traditional psychedelic molecules or less common ones.
Right.
Can you talk a little bit about how that influences psychedelic research? And I know you have sort of a story about how DOI has facilitated some.
Yeah, it's not exactly clear. This is a multi-year process that the DEA goes through to schedule. And these recommendations are made to DHS several years in advance based on, I'm not quite sure what data. I'm not, the transparency is not that clear on the decision-making on this. It's for example, for DOI, there's been discussion of scheduling that for over 10 years. And this is the second sort of significant attempt that they've made at scheduling DOI. And it sort of just falls into the category of, well, it's a drug that can be used for non-medical purposes, therefore it can be abused, and all drugs that can be used for non-medical purposes have to be scheduled. And psychedelics are in this category where there's at current no known medical use for psychedelics, despite clinical trials, despite the research. There's no marketed psychedelic. So by and large, so that means every psychedelic is therefore Schedule I and could be scheduled. What the UK does is they have an analog act that basically, if the structure predicts it's psychedelic, it's Schedule I. Here, they don't have it that strict, but if they see that people are abusing it, then they can recommend it for scheduling, which is what's happened with DOI and several tryptamines that are going on now. Is that they say they have data that it's being abused, and so they've recommended for scheduling and evaluation and public comment. DOI, though, is a key chemical. It's like the only chemical that is selective, it's known to be selective for 5-HT2 receptors, that has been used for the last 40 years almost. It was discovered in the early 1970s and has been used to study serotonin 2 receptor pharmacology for decades. Without DOI, I don't think any work really on 5-HT2A receptors would have been possible to look at their function. There are other drugs now, one that my father made called TCB2, which you can get from one vendor. But what research has shown recently is that not all drugs activate the serotonin 2 receptor the same. So having one drug that's available that activates the serotonin 2 receptor selectively is not really good pharmacology, because what we found is that DOI is very potent anti-inflammatory, but this other drug that this other vendor sells that my father had come up with, TCB2, it's a very good receptor activator, produces the same signaling, produces the same behaviors that DOI does, but it's not really anti-inflammatory. So it's activating the receptor differently, and LSD activates the drug differently. So to really fully explore what therapeutic benefit 5-HT2A receptors have, we need to have the tools that activate the receptor appropriately for these different disease models. Because when you just have a hammer, everything looks like a nail. And with the different drugs that are activating the 5-HT2A receptor, they're doing it differently. And by really restricting the tools available to pharmacologists, you're restricting the amount of knowledge that we're going to be able to extract from the pharmacology experiments to really identify what the potential therapeutic uses could be.
So this will be the last serious question here. We're coming up on the end of the hour. So blue sky, where do you see the field in 25 years or so? So at the moment, there's a lot of emphasis on sort of modifications of known compounds. They're a little bit modest for psychiatric indications, polymorphs of psilocybin for depression or whatever. What do you think will be the main story in 25 years? Entirely novel molecules, indications outside of psychiatry, or psilocybin for depression? I mean, where do you see it going?
25 years from now, what I would hope to see, pie in the sky, blue skies, would be primarily new chemical entities. I think, assuming that DOI and other tools aren't scheduled and made difficult to use by researchers to study basic fundamental science, we'll get an understanding of maybe if you activate the serotonin receptor in a particular way to emphasize one pathway, maybe that's better to treat depression than it is better to treat OCD than it is to treat inflammation. So once the individual pathways that are leading to therapeutic effect from the serotonin 2A receptor are identified, which really none of them are. We know that 2 of them involved in behaviors, but it's still unclear if it's involved in therapeutic effect. But once we know what these pathways are, then I think we can design new chemical entities that will selectively activate those pathways that will be beneficial for disease treatment. And that may mean that for treatment of psychiatric disorders, you have to have behavioral, the hallucination, the ego dissolution, the trip, that may be necessary. So coming up with truly selective 5-HT2A receptor agonists that are only activating those behavioral pathways rather than the 20 or 30 other pathways that are being activated, those might be the ideal therapy to treat depression. And like for anti-inflammatory, that you'll be able to pick up an over-the-counter nasal spray, pill, transdermal patch with some very potent sub-behavioral psychedelic that's only activating the anti-inflammatory pathway. So I would hope that we would see several different new chemical entities on the market, potentially tailored for each of these separate psychiatric and non-psychiatric diseases. And outside of the brain, we've just looked at anti-inflammatory. There are several other processes outside the brain and the immune system that could be potential targets. It's just nobody has looked at.
Great. So I do end with a bonus human interest question. Do you have a favorite fictionalized depiction of psychedelics in a movie or a book or anything like that?
Ah, I do. I think it would have to be from the film adaptation of Fear and Loathing in Las Vegas, when you've got the two main characters, one played by Johnny Depp, and the other senior moment escaping my mind now, plays the Samoan, who are in the lounge at the casino in Las Vegas, and everything starts melting and the other customers start turning into lizards, and Johnny Depp is like,
I like it. Your father actually said The Matrix, believe it or not.
That's my favorite. You know, it's The Matrix. That's interesting.
That was not it was not my guess.
That's interesting. I wouldn't necessarily. Well,
I wasn't sure either, but I didn't push back. Okay, well, thank you very much. This was great. We've come to an hour. Really appreciate this on behalf of lawyers who are trying to figure out psychedelics in California and maybe beyond. We really appreciate you taking the time. So,
thank you for the invitation. It's been my pleasure.
All right, I'll talk to you soon.